Thank you, Dr. Jones. So, like she said, my name is Melanie White. I'm a senior in animal science and industry. And for about the past year, I have conducted 2 studies looking at the effect of a high amylase corn hybrid in feedlot cattle diets, specifically on the ruminal fermentation of starch. So first, it's important to note that corn is an integral and, in fact, the main carbohydrate source fed to millions of feedlot cattle each year. However, ruminants have limited pancreatic amylase secretions, alpha amylase being the main enzyme that degrades starches into simple sugars for absorption. So instead, they use ruminal fermentation to degrade the majority of starches that they consume. And so the question was posed that if cattle are provided finishing diets containing additional amylase via a high amylase corn hybrid, how would that impact rate and extent of starch digestion? In these studies, we utilized Inogen feed corn from Syngenta seeds as our high amylase corn hybrid. In both studies, we used 7 multi-cannulated Holstein steers in a replicated crossover design. What this means is we randomly assigned each steer the Inogen feed corn diet or the control corn diet, which then alternated throughout the 4 feeding periods in each study. Now, the first study used diets containing a mixture of high moisture corn and dry rolled corn, while the other study utilized diets containing steam flaked corn. These diets were fed for an adaptation period of 9 days, with incubations occurring on day 10. Incubations involve Dacron bags filled with 1 gram of control ground corn, put within a mesh bag and then into the rumen. Now, these bags were removed after a predetermined amount of time, either 0, 4, 8, 12, or 16 hours. Once removed, they were dried in an oven and then weighed, and the resulting data was analyzed using proc mixed in SAS for percent disappearance. As you can see, we have 2 different graphs here. On the left, we have our 0-hour corrected percent disappearance with diets containing high moisture corn. And on the right, we have 0-hour corrected percent disappearance for diets containing steam flaked corn. On both y-axes, we have percent disappearance in dry matter. And on our x-axis, we have ruminal incubation in hours. The red line denotes results from control diets, while the purple line denotes results from Inogen diets. So first, we looked at the interaction of the effective grain on the effective hour and found a p-value of greater than 0.05, meaning these results had no impact on each other. Then we looked at the effective grain and found a p-value of greater than 0.05, which indicates no statistical significance. So this means that on both diets, percent disappearance was roughly the same. Then we looked at the effective hour and found a p-value of less than 0.05. So this was a statistically significant result, showing that as time progressed, percent disappearance increased on both diets. To summarize that a little bit, the steers on control-containing diets and the steers on Inogen-containing diets fermented starches at similar rates and to similar extents. This ultimately means that finishing diets containing this alpha amylase corn hybrid did not impact ruminal fermentation. However, this does not eliminate the possibility that these diets had impacts elsewhere in the gastrointestinal tract or could have impacts elsewhere in the realm of cattle nutrition, such as perhaps feeding a transition diet. Finally, I would like to thank Syngenta Seeds for sponsoring this project. I learned so much about the world of research and cattle nutrition, and I'm so excited to learn more as I go into graduate school. And I would also like to thank Dr. Kim and Mark Young for sponsoring this scholarship. I was so excited when I received the email, and I'm still so excited to for this honor. So thank you.